Okay, so for our group, we're looking into the genetic difference between HIV1 and 2 that lead to the difference in their transmission rate and disease progression.
Okay, for the introduction, HIV is a kind of virus that attacks the cell of the human body that fights infections. This would normally be white blood cells or T lymphocyte with CD4 receptors. We are mainly focused on the two major types of HIV. There are HIV1 and 2. There are many similarities between the two, including their basic gene arrangement, the way of transmission, intracellular replication pathways, and they're both resulting in AIDS. However, HIV2 is different in that it has a lower chance of transmissibility and has a less chance of progressing to AIDS because it needs a higher CD4 count to progress. So our hypothesis is that there should be a mean differentiation in the genetic coding that can explain the difference in transmissibility and the rate of disease progression. For our method parts, we use the NCBI blast to determine the final genetic relationship between HIV1 and 2, and we also use it to determine the genetic difference between HIV1 and 2. We use NCBI gene search for unique genes for HIV1 and 2. And we also use UniProt search for the gene function analyze. And that's it.
Okay, so then looking at a few of our early results, here we have two sets of phylogenetic trees. The one on the right is a BLAST result, which is focused only on HIV1 ref sequence against HIV2 ref sequence, all in relation to simian immunodeficiency virus. And so this what this one on the right tells us is that there's an actual phylogenetic relationship is hard to determine because it's basically showing that HIV1 is HIV2 is an ancestor of HIV1. And then on the left, we see one that phylogenetic tree that is a independent study, which was focused on the origins of HIV. And it shows that there's actually independent evolution of the two strands of the virus. So then moving on to this phylogenetic tree. This is a ref sequence of HIV1 against HIV2 without the limitation of using the ref sequence of HIV2. It shows HIV1 as a descendant of HIV2, and there could be a sign of cases of infection in which both HIV1 and HIV2 strains are present, leading to a new cross species. Finally, looking at the gene search of HIV1 and HIV2 and comparing them together, we see that there are a lot of shared genes between the two strands. However, HIV1 has 3 different genes that are only unique to HIV1 and 1 gene unique to HIV2. And we'll talk more about that in the next slide.
Yeah, so in specifically HIV1, we have 3 unique genes, which are the GAG, VPU, and ASP gene. Both GAG and VPU participate in the role by helping with the viral budding, while ASP, unfortunately, there isn't much study about the gene itself, so its function in HIV1 is still unknown. For HIV2, it plays a major role in suppressing the host's innate immune response, and it's accomplished through degradation of host SAMHD1 enzyme. So in conclusion, so based on the BLAST result, using the HIV1-2 reference sequence, we learned that HIV2 is an older lineage of lentivirus than HIV1. However, through literature review, we found that this is inaccurate description of the relationship. In reality, HIV1 and 2 are involved independently. Using the NCBI gene search, we found that HIV1 has 3 unique genes, while HIV has only 1. I mean, HIV1 has 3 unique, yeah. The HIV2 VPX genes plays a role in suppressing the host's innate immune response. And by having this function, it lowers the progression, lowers the human's immune response against HIV virus, and that leads to a slower progression towards AIDS. Well, for the HIV GAG and VPU genes, they plays a part in the viral budding process, and because of it increased the viral budding, we suspect that this lead to a higher concentration of new HIV virus made and also export out of the infected cells. And because of the high concentration of HIV, it will lead to repetitive infection within the host, and also a faster progression towards AIDS. And this high concentration also lead to the fact that if there is a chance of transmission between 2 humans, the chance of transmission will also be higher when it compares to HIV type 2. So we also learned that HIV1 and 2 independently involved. They're unique genes that lead to the difference into the transmission rate and the disease progression. For future study, we think that our study will help to understand the HIV lineage and also the evolution of HIV virus in the future. And this project and also bring us some insight into possibility of new HIV lineage in future.